what's going to happen is it inactivates it. And that's just because when blood clots, you're going to find that it takes a whole cascade of reactions to occur, and if any one of them doesn't take place, the blood's not going to clot. It's the reason that we have a cascade of reactions take place. It's ensuring that we're clotting what needs to be clotted. Okay?
So hemophilia, is that like generic or like
genetic?
Not generic, genetic. Is it genetic and it is it missing like a fibrinogen or is it just missing a clotting factor?
A clotting factor. Okay. So what's going to be missing? The hemophilias, because we've got different types, like hemophilia A, B, that sort of thing, that's going to determine which of those clotting factors is absent. And it's going to determine the severity. Because, you know, some people can have hemophilia and they still pretty much go along with life. They just know that, like, if something happened and they got cut or whatever, they probably need to get to a hospital to get the clotting factors to stop the bleed. Okay? Other times, people can have hemophilia so severe that something as simple as that, just simply walking or just bumping into something, causes them to bleed all underneath their skin, go to the hospital. I mean, it's a huge, it depends on the severity.
Is that usually in older people?
No, hemophilia is hereditary, so people who have it are going to be born with it, and they don't, you know, if you know that it runs in your family, you can, of course, have the genetic testing done before birth to know, okay? And if you've got enough money, you can have the child, you can have that genetic, where they take the egg and the sperm and they make sure they remove that gene, the genetic engineering. And then you use the, you know, in vitro fertilization so that your child is not born with it. Okay? So it's usually done from birth or before. Now, other proteins that we're going to have, immunity. That one's huge. We're going to continue, even though we're going to talk about some of it in this chapter, when we get to chapter 21, I think it is, we're going to be continuing with immunity. I need proteins for transportation. The last chapter that we did on hormones talked about the ones that would get to flow freely in the blood, but some were attached. And they were attached to a transport protein. So in my blood, I have proteins that act with clotting, immunity, and transportation. The categories that exist for these proteins: albumins, globulins, and the fibrinogen. So, proteins in our blood are important. The albumins. Ever heard of this one before? Yes. So you will hear it referred to in, like, the egg white. Albumins, okay, as a whole, they are the most abundant in my plasma protein. Their job: transport solutes and help buffer the pH of my blood, making sure that my pH of the blood in the range of roughly 7.35 to 7.45 is extremely important. Now, if we think 7.35 to 7.45, let's say that I went from 7.45 to 7.55. Is that a problem?
Yes.
Yes. And here's the reason why. The pH scale. Seven, neutral. Anything off to this side is base. Anything off to this side is an acid. Blood should be roughly falling about right there. Acids are based on a H plus, a hydrogen ion. A base is a hydroxide, an OH negative. So if I'm in a solution and my pH is neutral, I have the same amount of positives and negatives. Is everybody following me? Now, blood, okay, is just a little smidge over that 7. So let's add one more. Okay, so now I'm just a smidge of the negative over the positive. So everybody see how I did that? Well, if it goes from the 7.45 to 7.55, do you happen to remember what the increase in our ions is based upon? Is it a linear scale or is it logarithmic? It's logarithmic. So that means any increase is based upon a factor of 10. So if I went from 0.45 to 0.55, I have done a 10-fold increase in my negatives. So let's say I have 5 now, and if I do a 10-fold, how many negatives do I now have? 5 times 10?
50.
I now far outweigh my positives. I cannot have that in my blood. I need to buffer that. So whether acidic or basic, any change that occurs as 10-fold. And that's bad. Okay? Really bad.
So when they test your albumin level, that's basically testing how well you would be able to buffer.
How many are present? Because there should be a count that should be present. And your norm, there's a norm for it, and it should fall within the norms. So if that's the case, then we know that we've got enough albumins to carry out their functions. Okay? Does that make sense? Now remember, in my blood, let's draw a blood vessel. My blood is the mixture of those red blood cells, white blood cells, and my matrix, which is my plasma. Okay? So, That means in this blood vessel, things are getting to keep their properties. So an albumin, which is a protein, do you guys happen to remember the structures of proteins? Primary, secondary, tertiary, quaternary. Primary structure is the stringing together of those amino acids. Secondary is when that begins to either create a double helix or fold. When that double helix or the folding begins to wrap in on itself, that's tertiary. And the folding or the helix was secondary. So the secondary folding in on itself is tertiary, and two or more tertiaries put together is quaternary. So proteins and albumin, all right, to my knowledge, albumin is simply the tertiary structure. So that means I have an amino acid chain that has folded in on itself. So that means that that albumin is keeping its shape. They are the most abundant. So I got a bunch of them. Does everybody? Does that make sense? Meaning, in my blood, this is getting to fly around. Okay? Therefore, having this cell, because that's what it is, okay, it contributes to something called viscosity. What does it mean to be viscous?
Thick.
How thick, the flow, that sort of thing. And my osmolarity. What is that? Now, that's my osmol. However,
Is it how much water can penetrate?
let's think about osmosis. Water. Think about this for a second. Our planet as a whole is about 75% water. Our body as a whole is about 75% water. A cell itself is about 75% water. And my blood is about 75% water. Coincidence? I don't think so. So now, if viscosity is based on how easily something can flow, how viscous it is, and then osmolarity, osmosis, based upon the water, and then if they contribute to it. If I have bunches and bunches and bunches and bunches and bunches of these, do you think my blood would flow easily, meaning the viscosity? No. So if my albumins are high in number, and I'll draw one of the little proteins, okay, that makes my viscosity drop. So if I have a whole bunch of these in number, what about the movement of water, the water content of my blood?